एक बार फिर आपका मैं स्वागत करती हूँ ये बेसिक जेनेटिक्स के क्लास में, जो जैसा कि आप अभी तक देखे होंगे कि 3 टीचर्स शेयर कर रहे हैं और यहाँ पर मैंने अपना पूरा एड्रेस दे दिया है और मेरा फ़ोन नंबर है और ईमेल एड्रेस है। यही फ़ोन नंबर पे व्हाट्सएप भी है, तो आप जब भी चाहें आप मुझे कुछ भी क्वेश्चन, अगर मान लीजिए मैं कुछ क्लास में पढ़ा रही हूँ, आप लोगों को अगर लगे कि नहीं मैंने शायद गलत बोला है, ऐसा हो सकता है ना। ऐसा कहीं ज़रूरी नहीं है कि टीचर हमेशा सही बोलता है, हो सकता है कहीं मुँह से कुछ गलत निकल गया हो। अब बिल्कुल अब बेहिचक आप मुझे बताइए कि मैडम यहाँ पर आपने गलत बोला है, शायद हमने कुछ और पढ़ा है, तो मैं एकदम अपने आप को करेक्ट करूँगी, मुझे बहुत अच्छा लगेगा। और कुछ भी डाउट है आप जस्ट कॉन्टैक्ट मी। So this is the 4th class of this week and we started talking about extranuclear inheritance and we talked about cytoplasmic inheritance, and then in this class we will come into another topic or we will discuss another topic, and that is infective inheritance. Now we take another example over here, and that is a mutation in the chloroplast in again Chlamydomonas. Okay? This is an algae and we know that in this algae there are certain species which show resistance to streptomycin. So, what is streptomycin? That is this is an antibiotic. So, this is an antibiotic. So, they show streptomycin resistance, and this was studied by Ruth Sager in 1954. So, as I was telling you, they do not have male or female, but they have plus strain and the minus strain. So, you have 2 mating types. MT stands for over here mitochondria. So, though mitochondria hai, mitochondria coming from the plus strain and mitochondria coming from the minus strain. Both of them they contribute equally. This is what we call as biparental. This cell and this cell, both of them contribute equally. So, you have equal contribution from both these cells, and however, the phenotype will depend upon the plus strain. Mitochondria coming from the plus strain will again dominate the phenotype. So, this is, though you see biparental inheritance, but still you see that the phenotype depends only on the plus strain. So, in a cross between streptomycin resistant mitochondrial plus and streptomycin sensitive minus, half of them are mitochondria plus, MT plus, half of them are minus, but the phenotype, all the progeny of the phenotype is streptomycin resistance, because the plus strain was streptomycin resistant, right. Now, in a reciprocal cross, if the plus strain is sensitive, you can see over here the plus strain is sensitive. So, if the plus strain is sensitive and the minus strain is resistance, this resistance does not come into the progeny. This is progeny. So, all the progeny are streptomycin sensitive. Okay. So, this means that the mitochondria that is coming from the MT plus strain, again it dominates the mitochondria from the minus strain. So, we see a kind of dominance over here, even if it is defective, it dominates, and then you can see the phenotype. Now, this is next example of what we call as Petite mutation. Petite का क्या मतलब होता है? Petite का मतलब होता है small, है ना। कोई भी व्यक्ति अगर हम बोलते हैं Petite, मतलब वो छोटा बच्चा है, बच्चा नहीं है, बड़ा आदमी है लेकिन छोटा, उसका कद काठी अगर छोटा है, स्ट्रक्चर उसका छोटा है, उसको बोलते हैं Petite. Petite एक इंग्लिश वर्ड है, ठीक है। So, it means small. So, another example is in Saccharomyces fungi. This was studied by Boris Efruzi in 1956 and his co-workers, and you can see these Saccharomyces cerevisiae, they form colonies. And what you can see is in the Petite mutation, the size of the colony is small, whereas in normal mutation the size of the colony is big and small. Okay? Now, there are few things written over here, but I will just go into it one by one. Okay. Now, again, as in other neurospora that we were discussing, they exist in two states, a haploid, that is N, and diploid, that is 2N. So, there are three different kinds of mutations that we are talking about of Petite mutations, because Petite is a small colony, this means small colony. So, this size of the colony may depend upon lot of different other genes also, right. So, one kind of Petite mutation, we call them as segregational mutation. What it means is that if you have cross a Petite with a normal phenotype and this is a zygote, this becomes diploid. So, these are haploid over here, and this zygote, it undergoes meiosis. So, when it undergoes meiosis, half of them remain Petite and half of them remain normal, that means the size is big. Another kind of mutation is a neutral mutation over here. So, this neutral mutation over here, I will just tell you कि क्यों हो रहा है ऐसा. Neutral mutation में what you see is the haploid cells are Petite and another is normal. Meiosis, this is 2N, this is a zygote, and when it undergoes meiosis, all these spores are normal. Okay? All these spores are normal. One thing you will notice over here, which is different from Neurospora, is that in Saccharomyces cerevisiae, after one meiotic division, you have 4 ascospores, whereas in Neurospora क्या हो रहा था, ये जो 4 meiotic products है, वो एक mitotic division they underwent a mitotic division, so you see 8. Here you see only 4. Now, in another kind of mutation, if you another kind of a cross, so this is called as neutral. Another is suppressive. Here what happens is, if the plus mating strain, ये plus है ये minus है, if the plus mating strain is small or Petite and if the minus strain is normal, zygote you have a diploid condition 2N, it undergoes meiosis and all these are Petite. All these are Petite. Now, what happens over here? Now, in this mutation, segregational mutation, where you see 50% normal, 50% pokey, this means that this kind of a mutant, this mutant phenotype is under nuclear inheritance. Okay? That means this phenotype over here, this is showing nuclear inheritance. Okay? 50% of them, because after meiosis you will have 50% minus, 50% plus, you will have minus and plus, and then both of them will be you will have Petite as well as normal. But here what is happening in neutral mutants, all are normal you are seeing. Now, why? How they are all normal? They are all normal because the mitochondria in the neutral mutation, which is coming from the plus strain, they are actually empty, they do not have anything. So, the mitochondria coming from the minus strain dominates and you see all normal. On the other hand, suppressive means that mitochondria which is coming from the plus strain, this is somehow dominating of the mitochondria that comes from the normal strain. This mitochondria over here, it has lot of mutations, and these mutations, they have a dominant effect. They have a dominant effect, and so therefore, you see all the progeny that is coming from the suppressive mutants is they are Petite. So, you have 3 different kinds of mutations which show Petite phenotype. They are segregational, neutral and suppressive. So, of this segregational shows nuclear inheritance because you are getting 50-50 over here. 50-50 progeny है, मतलब जो न्यूक्लियर डीएनए है वो डिवाइड हो जा रहा है, एंड ऑन द अदर हैंड जो न्यूट्रल और जो सप्रेसिव है जिसमें हमको चारों एक तरह के मिलते हैं या चारों दूसरे तरह के मिलते हैं, ये जो इनहेरिटेंस पैटर्न है, दिस इज बिकॉज ऑफ द मैटरनल cytoplasm or the maternal contribution or the contribution from the plus. So, this is an example of Petite mutation. Now, we come to another topic and that is infective inheritance. What do you mean by infective inheritance? मतलब कि somehow जो ये inheritance pattern है वो किसी तरह का एक infection cause कर रहा है. We will discuss what exactly I mean with it. And this is very well studied in Paramecium. So, Paramecium you all know, okay, it is a flagellate organism, it has flagella. Now, in Paramecium there are certain particles which we call as kappa particles, This kappa particles, it is now known, it secretes certain kind of toxic substance which we called as paramecin, and this paramecin or the paramecium which contains this paramecium or the kappa particles, they are the killer strains. That means they secrete the toxins and it kills the sensitive strain जिसमें paramecin नहीं है. So, you have killer strains and you have sensitive strains. So, killer strain because they have kappa particles, killer strains because they have kappa particles, sensitive strain because they do not have kappa particles. Kappa particles, they proliferate in the cytoplasm and they contain DNA and protein. We many think that these kappa particles are actually virus like particles. Okay. Now, however, the important thing over here is that the toxic nature of this kappa particles or the paramecin depends upon a dominant allele K of the nuclear DNA. So, अगर nuclear DNA में एक dominant allele K है तो वो kappa particle killer बन जाता है, लेकिन अगर एक paramecium है और उसमें dominant allele नहीं है, खाली ये रिसेसिव अलील से होमोजाइगस कंडीशन में, तो ये जो है पार्टिकल और ये जो पैरामीसियम है दैट विल नॉट फंक्शन एज किलर स्ट्रेन. सो फॉर द कप्पा पार्टिकल्स टू फंक्शन and kill and produce paramecin, dominant allele K is very important, right. Now, how this infection is transmitted to progeny during conjugation? So, as you all know that इनमें कॉन्जुगेशन होता है पैरामीसियम में. सो यहाँ पर हम दो तरह की कॉन्जुगेशन की बात करेंगे. एक है शॉर्ट ड्यूरेशन कॉन्जुगेशन जिसमें साइटोप्लाज्मिक एक्सचेंज नहीं होता है, ये करीब 3 मिनट के लिए ये पैरामीशियम जो है ये एक साथ रहते हैं. उतनी देर में क्या होता है, सो व्हाट यू कैन सी ओवर हियर इज ये किलर स्ट्रेन है, ये सेंसिटिव स्ट्रेन है. जब कॉन्जुगेशन हो रहा है and you can see this is the genotype is capital K and this is small k, and when they come together during conjugation in short duration, first only the dominant allele or let us say the DNA is transferred, DNA का ट्रांसफ़र होता है. ये जो ड्यूरेशन है that is only enough for the transfer of genetic material. That is only the time is इतना ही टाइम के लिए वो एक साथ रहते हैं जब जेनेटिक मटेरियल जो है एक सेल से दूसरे सेल में चला जाता है, एक पैरामीशियम से दूसरे पैरामीशियम में चला जाता है. तो जब साइटोप्लाज्मिक एक्सचेंज नहीं होता है तो जीनोटाइप ऑफ दीज आर कॉल्ड एज एक्स कॉन्जुगेट्स. तो जो जीनोटाइप है वो तो हेट्रोजाइगस हो जाता है, लेकिन कप्पा पार्टिकल्स का मूवमेंट नहीं होता है, क्योंकि कप्पा पार्टिकल्स आर प्रेजेंट इन द साइटोप्लाज्म. सो इन दिस शॉर्ट ड्यूरेशन द ट्रांसफर ऑफ और देयर इज नो साइटोप्लाज्मिक एक्सचेंज. साइटोप्लाज्म का आदान प्रदान नहीं होता एक दूसरे सेल में, इसलिए वो जब सेपरेट होते हैं खाली वो हेट्रोजाइगस हो जाते हैं, लेकिन साइटोप्लाज्मिक एक्सचेंज नहीं होता. सो एज अ रिजल्ट you have a killer strain, you have a sensitive strain. Why killer and sensitive? Why sensitive over here? Because here if you have, just understand the genetics. So you have capital K, small k, and when they divide, so division जब होता है, so you will have one of the paramecium receiving K, then it makes its copy K, and this will become small k, small k. Now, though they had kappa particles, but because they do not have a dominant allele, so they remain as sensitive. So, this is very important for you to understand that this, when the cell divides, the alleles capital K and small k they separate. When they separate, they will replicate. जैसे कि पैरामीशियम में आप लोग पढ़े होंगे मैक्रोन्यूक्लियस माइक्रोन्यूक्लियस होता है, तो मैक्रोन्यूक्लियस फिर दो होते हैं उस तरह से. तो ये जब वापस जब रेप्लिकेट करते हैं तो ये कैपिटल के हो जाते हैं, एंड दिस विल बिकम स्मॉल के. नाउ दिस वाज अ किलर स्ट्रेन. सी व्हाट यू शुड सी ओवर हियर, ये किलर स्ट्रेन है, लेकिन इसके जो दो डॉटर सेल्स होते हैं उसमें से एक किलर हो जाता है, एक सेंसिटिव हो जाता है, एंड द रीजन बीइंग दैट they have inherited the small recessive allele. So, therefore, the kappa particles they cannot proliferate, and so they remain as sensitive, and this will remain as sensitive because there was no kappa particles that was inherited. So, though they are capital K and small k, they remain as sensitive. So, you have 3 sensitive and 1 killer. In the next condition, when there is a long duration conjugation, you have cytoplasmic exchange हो जा रहा है. तो यहाँ पर अब क्या हो रहा है, जब cytoplasmic exchange हो जा रहा है, so इसमें भी वो कप्पा पार्टिकल्स आ जा रहे हैं, इस वाले सेल में भी कप्पा पार्टिकल्स यहाँ से यहाँ पर ट्रांसफ़र हो जा रहे हैं. तो जब कप्पा पार्टिकल्स यहाँ से इधर ट्रांसफ़र हो जाते हैं तो क्या होता है कि जीनोटाइप इज हेट्रोजाइगस. ओके, ओवर हियर कप्पा पार्टिकल्स are now in both because there is cytoplasmic exchange. So, they both become killer. In contrast to the previous one, what you see in this generation there was one killer, one sensitive, because the sensitive strain did not receive any kappa particle. लेकिन यहाँ पर अगर आप देखेंगे तो ये दोनों जो है ये किलर स्ट्रेन बन जा रहा है. तो अगर ये दोनों किलर स्ट्रेन बन जा रहा है, उसके बाद जब वो डिवाइड करता है, देन दिस बिकम किलर, दिस बिकम सेंसिटिव. व्हाई सेंसिटिव? बिकॉज अगेन दिस इज स्मॉल के. सो अगेन व्हेन दे डिवाइड, सो वन रिसीव्स के, इट मेक्स अनदर के कैपिटल डोमिनेंट allele. This receives a recessive allele which will make another recessive allele. So, both have paramecium, both have kappa particles, but because this is recessive, this remains as sensitive, this remains as over here sensitive. So, you have 50% sensitive and you have 50% killer. In the third generation, when there is cytoplasmic exchange. On the other hand, when there is no cytoplasmic exchange, you have 25% or 1/4 of the population which is killer, and 75% or 3/4 of the population which is sensitive. So, this kind of inheritance, we call this as infective inheritance, because this does not depend upon mitochondria or chloroplast, but it depends upon the particle, virus like particles that are present in the cell, in these cells, right. Now, infective inheritance is also observed in Drosophila. Okay. This is an example of Drosophila. We have been talking about Drosophila, and I am sure you all must have heard of Thomas Hunt Morgan. He was working in 1900s, early 1900s, and he worked on Drosophila. He was the one who showed linkage and sex linked inheritance, and infective inheritance is observed in Drosophila, and this is by the sigma particle. Now, what is this sigma particle? It has been observed that different groups of Drosophila, they respond differently to CO2. What is CO2? Carbon dioxide. Okay? And some of the Drosophila, it was observed, they are sensitive to carbon dioxide, some are resistant. So, you have sensitive and resistance. What it means is that when it is sensitive, it cannot tolerate increasing amounts of carbon dioxide. तो अगर कार्बन डाइऑक्साइड का लेवल अगर हम बढ़ाते रहते हैं धीरे धीरे, तो जो सेंसिटिव वाले होते हैं वो क्या होता है, दे बिकम पैरालाइज्ड बिकॉज द न्यूरॉन्स आर नाउ नॉट फंक्शनल. द न्यूरॉन्स आर नॉट एबल टू टॉलरेट द इंक्रीजिंग अमाउंट ऑफ कार्बन डाइऑक्साइड, and if this goes on increasing, so if the levels of carbon dioxide goes on increasing, then they finally it becomes fatal. Fatal का मतलब है they die. These Drosophila, they die when the CO2 concentration goes on increasing, and if this is the frequency. Okay. So, it was observed that this sensitivity to or resistance to carbon dioxide is because of certain particles which are called as sigma particles. Okay. So, a cross between a sensitive female and a normal male produces all sensitive F1. यहाँ पर अगर आप देखेंगे ये sensitive female है, ये normal female है. So, here you see this is a sensitive female. Now, this is a heterozygous situation and all of them are sensitive because the mother is contributing to the phenotype. And in this case what has happened, that mother is normal, okay, father is sensitive, but all of them are normal. So, what important thing to note over here is that the genotype of these two progeny both are same, both have capital S, but this becomes sensitive, this becomes resistance, this becomes sensitive, this becomes resistance. Why? Because the inheritance is coming from the mother. So, mother, if mother is sensitive, it does not matter what is the genotype, of the progeny. It does not matter what is the genotype of the progeny. What matters is what information is being transmitted from the mother to the child. So, this is what is infective inheritance, and this also follows a maternal effect from the mother. Okay. So, this phenotype depends upon what kind of mother is present. If mother is sensitive, all the progeny will be sensitive. If mother is mutant, mother is normal, all the progeny will be normal. Now, what are these sigma particles? So, these sigma particles, by using electron microscope, again a virus like particle is present. So, this virus like particle is responsible for giving this phenotype, similar to what was observed for Paramecium. So, the kappa particles and these sigma particles, they have, so you have kappa particles and sigma particles, they both show infective inheritance and they are virus like, and they transmit the information from the female parent to the progeny in Drosophila. In Paramecium, what we had seen that depending upon a mutant dominant allele K, the kappa particles will be active or they may not be active. So, this is about infective inheritance. So, I thank you all for attending this lecture. Thank you once again.